Okay, I think my presentation is visible to you.
Yes, sir.
Yes, sir.
Okay,
Yes, sir.
today we shall be discussing about BLAST and FASTA, two most commonly and popularly used softwares which are available online for doing sequence alignment. So this kind of sequence alignment is done for alignment of one to many sequences. That means if you have got one query sequence with you, that can be aligned with a large number of millions of sequences that are there in the databases to find similar sequences. So as you know that there are many databases in the public domain which provide information about various nucleic acid as well as protein sequences which have been uploaded to the databases by the authors around the globe. As we know, GenBank is such one single source from where most of the biological sequences are available. So we have to do a search to the databases to find similar sequences to query sequences that we find from our experimental laboratory. Suppose you have amplified a particular gene sequence of an organism, then you want to confirm the identity of the organism based on the conserved gene sequence that you have amplified by PCR. Now, for that, normally in case of bacteria, for identification purpose, we take into account the amplification of 16S ribosomal RNA gene of the bacteria by using some universal primers. That means these primers are applicable for all different types of organisms. If your organism is unknown, you do not know to which species it belongs, then you can use an universal primer for amplifying the 16S ribosomal RNA gene. Then the sequence obtained after amplification by PCR, either by sequencing done in your laboratory with a gene sequencer or by outsourcing, when you get the sequence, then that sequence can be uploaded to nucleic acid sequences databases to find similar sequences in databases. And if you find that there are some sequences showing very high level of similarity to the extent of 100% or near to 100%, then you can expect that your unknown bacteria also belong to that particular species to which the known sequences in databases belong. So this is called database search for finding similar sequences, or simply we say that this is sequence searching. So for this kind of sequence searching, two most commonly used online tools are the BLAST and FASTA. So they are normally used to identify homologous DNA or protein sequences. It can be used for both identification of homologous DNA sequences as well as protein sequences. They usually provide facilities for comparing the DNA and protein sequences with the similar sequences which are already deposited to the databases. So both of them follow a kind of algorithm that is usually referred to as heuristic algorithms. So these heuristic algorithms are not only applicable in computational science or in case of bioinformatics, but these kind of algorithms are usually used for many other branches of material sciences for certain specific problem solving purposes. So these heuristic algorithms are the ones that are designed to solve a problem in a faster and more efficient fashion than traditional methods by sacrificing optimality, accuracy, precision, or completeness for spread. That means there are certain algorithms which do a problem solving exercise, but in that process, we may lose some of the accuracies of the particular conventional method that is normally used for similar purposes. Okay, so purposes to develop these heuristic algorithms are to find the results within a short period of time very rapidly. Say, as I already said, that for finding the optimal similarity or alignment among sequences, the best method is to use the dynamic programming, as we have already explained. But dynamic programming methods are usually slower, and it cannot compare one query sequence to million numbers of sequence within a short period of time. It will take a long time. So that is why, to make it faster, certain other methods are used. Some other algorithms have been proposed which can find more rapidly the similar sequences, sacrificing in the process the optimality, accuracy, precision, or completeness of the search. So, in dictionary meaning, heuristic means enabling someone to discover or learn something for themselves. So, in these two softwares also, the accuracy is little bit compromised so that we can get the aligned sequences from among millions of sequences present in the databases within a short period of time. Let us today discuss about how they work. So, as I said, they are the software tools for rapid pairwise sequence alignment. So, normally they are used for aligning one to many, means out of millions of sequences in the database, one by one, all sequences will be picked up and they will be aligned to our query sequence very rapidly so that we can get the most similar sequences out of all the sequences in the database. So these software usually work by finding short stretches of identical or nearly identical characters. So, say for example, suppose each of the nucleotides will, or each of the amino acids in a sequence will be counted as alphabets, letters, or we can say character. It's a character, A, T, G, C. These are 4 characters in the nucleotide database, also nucleotide sequence. So a combination or a specific pattern of 2, 3, 4 nucleotides can form a word. So system will have randomly designed different patterns of words, means a word containing more than one character in a row. So similar kind of word formation by randomly picking up the different nucleotides will be generated by the system, and the sequences in database will be searched one by one for finding similar words in the database sequences. That means, first of all, the query sequence is divided or broken down into certain small pattern of nucleotides called words, and the system will search for similar kind of words present in the sequences of the database. So that means our assumption is that the two related sequences must have at least one word in common. Some commonality must be there in terms of words between the similar sequences. Now, first, as I said, they will identify the matches in the words and then extend it to the entire length of the sequences or regions of similar words, ultimately to get a longer alignment between the sequences. So that means first you will find out this exact identity in words in short stretches of the sequence, then it will extend the regions of similarity to the entire length of the sequences. Finally, it will align both the sequences one to other. So the adjacent high scoring regions are in the process joined together, ultimately to get a complete alignment. But there is a basic difference between BLAST and FASTA. While BLAST is mostly involved in finding an ungapped, locally optimal sequence alignment, the FASTA finds similarities between less similar sequences. So in the BLAST, usually ungapped or sequence pattern without gap is first formed as words, and they are found in the database sequences for finding locally optimal sequence alignment. But in case of FASTA, similarities are usually searched between less similar sequences. Now let us first discuss about BLAST. It extends as Basic Local Alignment Search Tool. So this particular search tool was developed by scientists and it is available for users through National Center for Biotechnology Information. It is present in the website of National Center for Biotechnology Information, or NCBI. The address is blast.ncbi.nlm.nih.gov/blast.cgi. So if you click on this link, you can reach the NCBI BLAST. So basically, there are 4 different types of BLAST available. They are nucleotide BLAST, protein BLAST, BLAST X, and T BLAST N. We shall discuss about them, how they differ from each other. So this was first proposed in 1990 by a scientist known as Stephen Elsul, and it uses the heuristic algorithm, as I already mentioned, so as to align one query sequence with all the existing sequences in a database, so that it can identify rapidly the regions of identity in terms of words by doing local alignment. So the system also scores a particular value, which is usually called as expect value or expectation value, or commonly referred to as E value. So E value is the number of expected hits of similar quality that could be found just by chance. That means sometimes the hits or similar scores between sequences can be observed by chance also, not by actual scoring because of their similarity. So the E value as an output given in the BLAST analysis will give you an idea in terms of probability how much it is probable to have an E value, means a particular hit or similarity quality or score by chance, not by real calculation. So that is why we always expect in a good alignment to have an E value as low as possible. If the E value is high, that means there is a high possibility that the similarity score is obtained only by chance, merely by chance, not by real measurement. Okay, so it should be approaching to 0. Whenever it is negligible, approaching to 0, or in a fragmented value, decimal value, it is always welcome, because that indicates that the E value is low, that is, the similarity score that is obtained is not occurring by chance, but it is the real score of similarity between the sequences. There are, as I said, different variants of BLAST. Although measured there are 4 variants, but we can say there are 5 different variants of BLAST. 5 different variants of BLAST. First one is BLAST N, or nucleotide BLAST. N stands for nucleotide. It is a BLAST or basic local alignment search tool used for similarity searching between nucleotide sequences. That means if your query sequence that you want to compare with the sequences of database is a nucleotide sequence, then we use BLAST N. That means we try to find out similar sequences in the nucleotide sequences of the database. Nucleotide searching against nucleotide database. Then similarly, there is a BLAST P. P stands for protein, means we compare the protein sequence with protein sequences in database. So you have a query sequence, which is a protein sequence or amino acid sequence, and you want to find similar sequences in the protein database. So protein against protein, that is BLAST P. Another variant is BLAST X. So in this case, it searches protein databases using a translated nucleotide query. That means you do not know what is the amino acid sequence or protein sequence coded by a particular gene sequence. But based on the gene sequence or the nucleotide sequence that you have, the system can predict the translated protein sequence, as I have shown in the last class. So in BLASTX, from a nucleotide sequence, first a translated protein sequence will be obtained, and that translated protein sequence will be blasted. So blast is also used as verb in that case. Blasted means search for similar sequences against the sequences in a protein database. So it is BLASTX means using nucleotide query sequence, you can find protein sequences of high similarity using the translated protein sequence obtained from the query sequence. Then tBLASTN. This is another variant of BLAST in which it searches the nucleotide databases which are translated and obtained in the form of protein sequences. So in the NCBI itself, there are databases containing protein sequences obtained by computational translation of nucleotide databases. So tBLASTN will use these databases to find out similar sequences against a protein query sequence. That means you have an amino acid sequence with you for analysis. You are using it for finding protein sequences in databases, but you are not using only the pBLAST or protein sequence BLAST, but you can also use tBLASTN, where it will also search for similar sequences in translated protein sequences obtained from nucleotide databases. Then last is tBLASTX. It again searches the translated nucleotide databases using a query sequence, which is also a translated nucleotide sequence. Okay, you have got a protein sequence. First, translate it to nucleotide sequence by using computational method, and that translated nucleotide sequence can be used as a query sequence for finding similar sequences in databases using those databases which contain translated nucleotide databases. Now, FASTA. FASTA stands for First All or FASTA, First A, or some people say First Alignment. So this is the URL or the website from where you can get the access to FASTA online tool. So this was the first database similarity search tool, which was developed earlier to that of NCBI. NCBI came later, but this was developed 5 years before BLAST. BLAST was developed in 1990, but this was developed in 1985 by a group of scientists, David J. Lippman and William R. Pearson. So the FASTA uses also the heuristic algorithm, as I explained earlier, but it uses a hashing strategy to find matches for a short stretch of identical residues with a length of k. The k may be anything, that means 2, 3, 4, 5. Okay, so these residues may indicate an amino acid residue or a nucleotide residue. It may represent one nucleotide or one amino acid. So different length of k, 2, 3, 4 nucleotides or 2, 3, 4 amino acids are grouped together like that of the words in case of BLAST, and they will try to find all these groups or the words in the sequences of the database. So these strings of residues in this case are known as k-tuples or k-tups. Instead of using the word, using the term word here, k-tuples or k-tups are used, and which are equivalent to words in BLAST, okay, but are normally shorter than the words. These are usually shorter than the words. They contain lesser number of nucleotides or lesser number of amino acid residues. Typically, the K2P is compared of 2 amino acid residues or 6 nucleotide residues. In case of protein sequences, all K2Ps will be composed of randomly coupled 2 amino acid residues. And in case of DNA, it will contain 6 nucleotide residues. And then the entire length of the sequences will be searched for presence of the K tuples. And then, as I said previously, in case of BLAST, after finding the number of K tuples in the entire sequence, they will be linked together to have the final optimal alignment. So, as the name itself indicates, as I said that in case of heuristic approach, we usually compromise the accuracy or optimality to some extent. Therefore, in both BLAST and FASTA, although we find the results very rapidly and we can have the sequence searching against millions of nucleotide or amino acid sequences in database within a short period of time, but we have to sacrifice the accuracy a little bit. So these methods may not accurately find the optimal alignment or true homologs. They are 50 to 100 times faster than dynamic programming. So this is the advantage of using heuristic algorithms rather than dynamic programming to have optimal alignment. But if you want to have accuracy in finding similarity between sequences, then it is always better to use dynamic programming methods. Thank you. So this is the theoretical aspect of BLAST and FASTA. And today we shall deal with the sequence alignment using BLAST. Practically, I'll show you how using the online BLAST tool, we can go for sequence alignment. Till now, if you have got anything to ask, you are welcome. Everything is clear to you?
Yes, sir.
Yes, sir.
Fine. So then let us go to the browser so that we can go to the online BLAST tool. So here either you can go to NCBI first, then download the BLAST homepage, or otherwise you can search in the browser directly the BLAST at NCBI. BLAST at NCBI. Okay, it will lead you to the downloading option or the real BLAST tool. Second option here, BLAST, local alignment search tool. So this is the homepage of BLAST. This is a part of NCBI. It is embedded within NCBI website. So here you see there are options here. The 4 important variants have been shown here: nucleotide BLAST, protein BLAST, then BLAST X and T BLAST N. So whatever you want to use, you can use. So you can use nucleotide. Let us first use nucleotide BLAST. Now for doing nucleotide BLAST, what will you require? You will require a nucleotide sequence. So already we downloaded some sequence in the last class. Have you kept it on your desktop? We can use those sequences.
Yes, sir.
Okay, so I'll go to my desktop then try to find out the sequences already downloaded in the previous class. First, we will take a nucleotide sequence. So, okay, sorry, it is open in BioEdit. Rather than opening in BioEdit, it will be better to open the sequence by using Notepad. So go to Open With, then click on Notepad. Right click, then click the option Open With, then click the option Notepad. So this is the sequence. So let us take one part of the sequence. Suppose entire sequence we are not taking. Only one part of the sequence that we have taken. This is the part of the sequence that I have. So this sequence I suppose do not know. It is a Escherichia STX1 already we have known sequence, but suppose this is a sequence that we have obtained from our experimental work in the laboratory by amplifying a gene. We do not know which bacteria it is or which gene it is. So this is a part of the sequence. Let us copy it. Okay. Then we will go to the BLAST, the home page of the BLAST. So here, now in this box, this query box, you have to enter the query sequence. Either you can write the accession number. If it is a downloaded sequence from databases, you know the accession number, or maybe a sequence you yourself have deposited to the database and they have given you an accession number, then you can put your accession number here. Or otherwise, you can put the entire sequence. Now we will put the entire sequence. Control V. We have pasted here. No need to make it contiguous. Let it be like that. Whatever way it has been presented, it should be like that only. Okay, keep it like that. Then we will use, in this case, for common BLAST, the database within NCBI called nucleotide collection, NRNT. This data, against this database, the nucleotide BLAST is commonly done, because we do not know to which particular database, sequence in the database, our query sequence will be similar. We do not know. This is the nucleotide collection where it contains all the nucleotide sequences of the gene bank. Only thing is that there is no redundancy or duplicacy, but it contains all nucleotide sequences, including partial sequences, complete gene sequences, complete genome sequences. So any kind of nucleotide sequence deposited to the gene bank will be available in this collection, what is called nucleotide collection, NR/NT. NR/NT means non-redundant nucleotide. Okay, this is a non-redundant nucleotide database. It means it's a nucleotide database containing all sorts of nucleotide sequences. Only there is no redundancy or duplication. Normally we select this by default. Then optimize for, you see, highly similar sequences, which is called mega BLAST. Then more dissimilar sequences. If you want to find more dissimilar sequences, it is called discontiguous mega BLAST. Another type is somewhat similar sequences. It is called BLAST N. Now normally in a common BLAST by default, we select highly similar sequences or mega BLAST. That means usually this sequence searching is done only to find out identical sequences, more similar sequences. So we will click by default highly similar sequences. Then we click on BLAST. After clicking on BLAST, this information of the query sequence will be now given to the NCBI database for similarity searching by using the heuristic algorithm. And you see, so fast, within few seconds, it has given us the result. It would not have been possible if the dynamic programming is used, because the nucleotide database of NCBI contains millions and millions of sequences. Against all of them, each one of them, it has done the sequence searching. And you see, within a short period of time, within very few seconds, although we are searching from India, the server is in US, but we could get the information within seconds. So fast it is. So this has been possible only because of using the heuristic algorithm. Okay. So you see, it has given us the sequences which show highest level of similarity in preferential order. In a priority order, it is given. That means the sequences showing highest level of similarity are shown first, then sequences with lesser. So you say so many sequences it could find where there is 100% similarity. You see here, maximum score is 265, total score 265. Total possible score is 265. Out of them, this sequence is showing 100% similarity. Query coverage is 100%, means entire length of the sequence is covered. Then percentage of identity is 100%. And the E value is 67 to the power minus 7e. What does it indicate? It is a e. It is again exponential value in mathematics. I think in mathematics you know what does it mean. 67 whole to the power minus e7 or 7e. That means before 67 you are to put 5 more 0, then before that you have to put the decimal. That means after decimal there will be 7 digits in this figure. This indicates. Okay, that means the e value is very less, negligible, almost zero. Means it is a very less chance that this sequence similarity or high score has been shown by chance, but this is the real score. So these are the sequences, because as we know that this is a STX1 is a common gene present in many pathogenic Escherichia coli. So already so many Escherichia coli complete genome sequences are uploaded or deposited to the NCBI data bank, which contains information about the sequence of STX1 also within it, because this is a part of the complete genome. So mostly when the strain is a O157H7 strain, all O157H7 strains usually contain this particular toxin, what is called Siga-like toxin or cytotoxin. So cytotoxin producing or verocytotoxin producing Escherichia coli mostly belong to O157H7 strain. So this is the serotype, and this serotype is more commonly involved with two important diseases in human being: hemorrhagic colitic syndrome, hemorrhagic uremic syndrome, and hemorrhagic colitis. Hemolytic uremic syndrome, hemolytic uremic syndrome, and hemorrhagic colitis. So these two diseases, HUS and HC, these two diseases are most commonly found in advanced countries or Western countries like US, Canada, etc., when human beings are infected with E. coli of O157H7 strain, because this strain contains a typical cytotoxic component in it. So that is capable of destroying cells. So they can destroy the enterocytes, the cells in the inner lining of the lumen of intestine. So, but normally, although we have so many E. coli having this particular virulence marker in our country, but this disease is not commonly seen in the population in India, because we are resilient to this infection. But this particular strain of E. coli is also responsible for a disease in pigs, which is called edema disease. Edema disease in pig. So similar cases of edema disease have also been reported from some parts of India, including Assam. Okay, you see these strains all belong to O157H7 and they are showing 100% similarity. Even there are many other strains not belonging to O157H7 which are also showing presence of this gene. These are all Escherichia coli. So this is the kind of sequence similarity that we have found. You see, all these are showing 100% similarity. So E. coli verotoxin subunit A, VTX1 gene means STX1 gene. Not only the complete genome sequences, but also it is showing similarity to the gene sequence also. So if we want, we can download these sequences from the GenBank by simply clicking on these accession numbers. You see, these are the accession numbers in the last column given. So here, if you click on the accession numbers, you can download the sequences of the database where our query sequence is showing 100% similarity. Okay, so this is one of Escherichia coli strain RM13322 chromosome. So this will be showing all information pertaining to that particular sequence, and it will also give you the sequences which have shown 100% similarity to our sequence. Okay, it's a genome sequence. It's not a gene sequence, it's a genome sequence. So it will be a very large sequence. So it is still being downloaded. You see, 3.23 MB. It will be more than that because it's a genome sequence. So like this way, we can go for NCBI BLAST against a query sequence, which is a nucleotide sequence. So this BLAST technique is very commonly used for many purposes in bioinformatics and for that purpose in molecular biology for finding similar sequences in the databases, not only for identification of the organism, but also to find the mutations that might have been reported in similar genes by doing the same kind of similarity search. If you find that some sequences in the database belonging to the same gene and same species of the organism are showing some variability in terms of similarity ratio, say some is showing 97% similarity, some ones are showing 98% similarity, then you can find out where they are dissimilar. That means where mutations have been already reported in some other strains. Although they belong to the same species, they may belong to the sequences may belong to the same gene, but there may be certain mutations at some places. So those mutations can also be found out. Similarly, for many other purposes in molecular biology, the sequence searching by use of BLAST can be done. So similarly, we can also go for sequence searching against protein sequences. So let us see how it is done. So for that, again, we'll go to the folder containing sequences that were downloaded. Already we downloaded protein sequences. Okay, this is my protein sequence, STM0171 protein. So I'll right click, open with Notepad. Then this is the protein sequence. Let me take a part of the sequence, not the whole sequence. Let us take, suppose, this part. Now this part is not known. This is a amino acid sequence that we want to compare to which particular protein it belongs. It's a part of a protein sequence, but to which protein it belongs, we do not know, suppose. Then let us find out by sequence searching. So I have copied it. Then this time we will go to BLAST P, not the nucleotide BLAST, but the protein BLAST. So we'll go for BLAST at NCBI. So under that, this time we'll select BLAST P, protein BLAST. Okay. So this will be searching protein databases using a protein sequence or amino acid sequence. So we'll give our input in this box. Then we keep it as database against which it will be searching, non-redundant protein sequence database. Here we will not be clicking nucleotide databases, but we need protein sequence database because we are going for a protein BLAST. And by default, the system will select this particular database, non-redundant protein sequence, NR database. Then we click BLAST. So similar to previous occasion, within few seconds, it will compare with the sequences in database, and rapidly it will give us the similarity search results showing the sequences showing highest level of similarity to the query sequence. Now you see, this sequence was from STM0171. That day we already found that ST010171 is the gene which code for an enzyme in Salmonella, which is called carbonate dehydratase. I think you remember. And this particular gene is present across different Salmonella serovars or serotypes. All Salmonella enterica serovars have got this particular gene in common. And that is why this is one of the common targets for identification of Salmonella enterica, or it is a internal control for many multiplex PCR reactions developed for identification of Salmonella organisms. It's an internal control because this should be present in all Salmonella. So you see now, therefore, it has shown 100% similarity, 100% cover query cover and 100% similarity with a e-value of very, very negligible 73 to the power minus E6. So that means the before 73 there should be at least 40, and before that only the decimal point will come. So that means it is almost 0. So E value is almost 0. That means it's a good data showing the accuracy in this code. It is not occurring by chance. So this is always hitting the carbonate and dehydratase protein in all the salmonella. In many different salmonella serovars or many different strains of the organism, it is showing 100% identity, but all are showing the identity with only one particular protein, that is carbonate dehydratase. You see, till the last, you see, all are showing 100% identity to only to one particular protein or enzyme, which is called carbonate dehydratase. That means you can say that if our sequence was not known, then we can immediately say that this is the particular sequence which code for carbonate dehydratase enzyme in Salmonella enterica organisms. So somewhere among the different similar isolates, you will, if you go to the real sequence by downloading, by clicking on the accession number, you may also find in the locus where it is matching the position in the genome sequence where it is showing complete match. Then you, from the genome sequence, you can also find what is the name of the particular gene. So by this way, the sequence searching by BLAST can give us lot of information pertaining to a query sequence of nucleotide or protein. And this platform also gives us the opportunity to download highly similar sequences for their sequence comparison or for use of those data for our analytical experiment using different bioinformatics tools. So in the process of using the different bioinformatics tools in future classes, we will be frequently using BLAST again and again, so as to show you how the BLAST is very useful in different biological experimentation done in silico. Okay, in bioinformatics, therefore BLAST is a very, very important and popular tool that is required to be done for many different purposes. So with this, let me stop for today. I think there is no question from you. If you have got any query, you are welcome